Hello and welcome to this special show on Rajya Sabha television. I am Vishal Dahiya, and here we take a stock of India's fight against COVID-19 pandemic. Now today we're going to talk about a different form of treatment for COVID-19 on which researchers have been working upon, that is monoclonal antibodies. What exactly are monoclonal antibodies and how do they work as far as COVID-19 is concerned? We'll try and understand all these and figure out how exactly is monoclonal antibodies helpful in COVID-19. For more on this, we're joined by a distinguished panel of guests. Let me first introduce them to you, beginning with Dr.Arvinder Soin, Chairman of Medanta Institute of Liver Transplantation in Gurgaon. We also have with us Dr. Pramod Julka, Senior Director, Max Institute of Cancer Care, and Dr. Vishwakarma, an advisor with CSIR, New Delhi. Welcome, all of you gentlemen, to Rajya Sabha television. Let me begin with you, Dr. Pramod, and let's start by giving our viewers an overview of what exactly are monoclonal antibodies.
Actually, these are antibodies which we make use of in other diseases also, like cancer. We are using monoclonal antibodies. For example, I will give you an example of breast cancer, where we are using these antibodies, monoclonal antibodies. We are using in lymphomas, blood cancers. We are using in various types of cancers, where there is immense growth of the cancer cells, and we subject, because every antigen in the body, makes an antibody. So antibodies are present in our body. So what happens? We make these artificial monoclonal antibodies against a particular target, against a particular antigen. So what they do, they attack the same antigen and then kill the cells. For example, immunotherapy we say in cancer also. Similarly, because in cancer there is immunosuppression. And in cancer what happens? Once we give immunotherapy, the T cells, our bodies has Army, Navy, Air Force, we call it, and that is lymphoid cells, lymphocytes. So our lymphocytes are T lymphocytes and B lymphocytes. So these T lymphocytes, they go and kill the cancer. So this is called immunotherapy, and we give there are breaks on the T cells, and we release the breaks and then attack the cells. Similarly, in monoclonal antibodies, there are certain antibodies which can lower the IL-6 levels, interleukin-6 levels, which are very high in COVID. COVID, there is inflammation. There is a lot of inflammation, and inflammation causes increase in IL-6 levels, interleukin-6 we call it, and interleukin-6 high levels, they can attack any part of the body, and most likely they attack the lungs when people fall into either comatose condition, or they land in ICU, or they land with pneumonia, or they land with certain, very intensive illnesses. So these IL-6 levels, for example, tocilizumab, this is an antibody, monoclonal antibody, which lowers the IL-6 levels. And once they are lowered, then we can tackle COVID-19 conditions very easily. They have to be used earlier and not once the patient goes in coma totally. Or patient goes into ICU and patient lands up in morbid condition.
So basically this is a form of treatment which is to be used in the early stage of a COVID-19 treatment there. Let me bring in Dr. Soin here as well. Dr. Arvinder Soin, from your point of view, if you would like to add a bit more to the, for our viewers to understand the concept of monoclonal antibodies, and also, the reason why we are exploring it as a treatment against COVID-19.
See, antibodies are substances, they are protein substances in the body against antigens. Now, whether it's a cancer cell or an organism of infection, they are defined by certain targets on their cell surface. It's like you have a name, I have a name. So they have a name, they are called antigens. And these antibodies attack those antigens either bind with them or prevent their binding to the important receptors or sites in the human body. So these antibodies basically prevent the interaction of the offending organism with the point in the body where they have to attach.
Okay.
So in COVID-19, these monoclonal antibodies are being directed against the spike protein, which everybody has seen in pictures. You've seen all those spikes on a round ball. So that's what the virus looks like. So at the end of that spike protein, this antibody will attach, and that will prevent this spike protein to attaching to the lung cells where it hurts the most, right? So the site where the coronavirus spike protein attaches is the ACE2 receptor, and this monoclonal antibody, the treatment, will prevent that attachment of the virus to the lung cell.
Okay.
So that's what a monoclonal antibody does. Now, monoclonal antibodies are typically mass producible. They can be artificially made. In the oldest method of making these artificially is basically in mice or animals. So I won't go into the details of how that is done, but basically from COVID patients, from the blood of COVID patients, you remove the antibodies, then you identify which is the one or the ones which are most effective against the spike protein. Then you mass manufacture them. And then you put them again into future patients, either early in their disease. So this antibody prevents the binding of the virus to the lung cell, or you can actually give them prophylactically in advance to high risk groups so that these antibodies will attach to the receptors, to the cells, and prevent the virus attaching in the future. But this effect only lasts a few months. Okay. So they can be used as treatment as well as vaccines. And that's the context of using them in COVID-19.
Okay, both as a treatment as well as vaccine, although the duration or the period will not be very long, as Dr. Soin is saying. Dr. Vishwakarma, I'd like to bring you in here. In terms of, how do you view this sort of new line of treatment, although we've been using, as both, Dr. Soin and Dr. Pramod is pointing out, that we've been using this line of treatment as far as other infections are concerned. But for COVID-19 specifically, how do you view in terms of its use there or utilization specifically in the early stage?
Yeah, so I think the concept has been described, but as I have been discussing with you over the months, we are now moving into a different phase for therapeutic for COVID-19. In the early few months, as you know, we were struggling, trying to repurpose both small molecules and biologicals. I think we had some mixed success. We had dexamethasone, and we had some positive data with tocilizumab. So that was the situation. Now we have moved into a very, very decisive new phase where now these next generation monoclonal antibodies are being designed for the virus. So far all those monoclonal antibodies were there, either they were autoimmune diseases or oncology products, and they were repurposed. And fundamental difference between monoclonal antibodies and something what we call polyclonal antibodies are that these monoclonals are very specific to a particular epitope of the antigen, while the polyclonal will have multiple epitope covered in one particular molecule. So sometimes they are more effective, sometimes monoclonals are much more sharper and more effective. So I think the most leading antibodies right now in clinical trials, advanced clinical trial, the Regeneron, you must have heard yesterday that the President of the United States has been injected by that experimental medicine. So that's specifically designed for COVID-19. So this is, I think I consider is a very decisive phase for drug development, where we are now getting these therapeutic monoclonal antibodies which are designed against the antigen and epitope of the virus. And there are two other which are very advanced stage. Yesterday data was presented for Eli Lilly program, which is happening, and another project with AstraZeneca. And all three projects are in now phase 2, phase 3 trials, and they are very scientifically sort of supported by NIH research program, which is something very good with these development program, where pharmaceutical industry, and like Dr. Sen said, that the one major advantage with monoclonal is that there is no limitation of how much you want to produce. So you must be hearing in the media that there will be shortage. So I don't see any problem you can produce, because basically they are immortalized by fusion with the cancer cells. They are hybridomas, and you can produce as much as you want in the laboratory. So I think that's the best part of these therapeutics biological therapeutics.
Okay, okay. But you spoke about, the data which is coming in from the evidence-based research on this, and I'd like to bring Dr. Pramod here on that aspect. Dr. Pramod, what are the initial results suggesting, of use of monoclonal antibodies vis-a-vis COVID-19? As Dr. Vishwakarma was also pointing out, all of us have heard this line of treatment being used on the President of the United States, but apart from that, because still it is being considered as an experimental form of treatment.
Yes, it is actually, as earlier said, monoclonal means mono means against one particular antigen, and we say, as Dr. Vishwakarma said, that against epitope of one antigen. So it is monoclonal means against one particular clone of cells. So these attack only the same target proteins or the target antigens, and then we get the results. So they are not polyclonal, they are monoclonal antibodies, and refined monoclonal antibodies which are being tried, and maybe in future we would try some more, because now vaccines are coming up. Every country is trying to make a vaccine out of these. So these monoclonal antibodies, once they are refined, you can make as many as possible. Earlier, as Dr. Soin also told, earlier they were made in animals, but now they are made in human beings. So we are using human monoclonals also. As I was telling you about because I being an oncologist, so I would talk about cancer. So we are using daily in cancer, chemotherapy, immunotherapy, all these, we are using monoclonal antibodies. So here also, against COVID antigen, that virus, this goes into the cells by attaching to ACE2 receptors. These are the receptor cells. And we want to prevent this virus entering inside. And there we attach the monoclonal antibodies, and then they attack either the virus or the proteins against which the virus attacks.
Okay.
So that is the use of monoclonal antibodies.
Okay. Okay. Dr. Soin, I'd like to bring you in here, and let's understand a bit of a concept here vis-a-vis monoclonal antibodies, because it'll be really wonderful for our viewers to understand. On this show, we've been talking about all form of therapeutic treatments and new ideas which are being worked upon. Initially, when convalescent plasma therapy was talked about, we discussed that as well. Anti-sera being developed. So what's the difference here when we talk about these different form of treatments? Monoclonal antibodies, the convalescent plasma therapy, anti-sera. How do they differ from each other in terms of obviously the use as well as their effect?
That's a very relevant question, Vishal. Now let's look at convalescent plasma. It has a lot of antibodies. Some of it may be specific to the virus, and some of it may be not. In fact, a lot of it is not. So if you've read the guidelines of FDA and the Ministry of Health and ICMR, there is a certain titer of antibodies, which means, let's say there are 100 units of antibodies in 1 unit of plasma, which I donate after COVID. Out of those, at least theoretically, let's say 10 or 20 units should be specific antibody, which will either attach to the virus or attach to the receptor, the ACE2 receptor, so that the virus attachment to the lung cell can be prevented. So if you put in lots of plasma and the plasma is not rich in those particular antibodies which are relevant in preventing the interaction of the virus with the cell, then it's not of much use. Whereas this obviously is monoclonal antibody, which means every single antibody in the solution that you infuse or give to the patient, each of those antibodies will be relevant. So 100 out of 100 units will be effective against the virus. So that's the big difference between convalescent plasma therapy, which is like a general infusion of plasma taken from a COVID patient who's recovered, versus specific monoclonal antibodies which have been manufactured just specifically for that virus. So, it's like a concentrate of antivirus antibody as opposed to plasma, which is like a general solution which has some effective antibody and some not.
And
so that's the major difference. The other thing is that No. So the anti-sera are just an extension of the same plasma that you're talking about. Basically.
Okay.
They all have varying amounts of specific antibody.
Okay.
And the most specific is a solution containing only the monoclonal antibodies, which is being tried at the moment. I mean, President Trump may have received it, but there is no conclusive scientific proof that it works at this point in time. I mean, it's being tested in the primates in the laboratory, and it's being trialed in phase 2 trials since August. And which basically means, I think the Regeneron guys are planning to give it to 2,000 patients. They started in the US, they started in the UK. So this trial will probably finish in the next month or 6 weeks, and then we shall have some results.
Okay.
But these results come out much faster than any vaccine results, because typically, for a vaccine result to come out to tell us whether it's going to be effective, it's going to take 20 to 30,000 patients. Whereas this only needs 2,000 or 3,000 patients to actually tell us if it's working or not.
Okay. But one more aspect here, Dr. Soin, and that is, as far as convalescent plasma therapy is concerned, repeated studies, and the latest one being conducted by ICMR showed that, there's not much significant effect on bringing down the mortality rates. So compared to that, when we talk about, monoclonal antibodies, can we hope that, this line of treatment might be a bit more effective than what we are talking about in terms of convalescent plasma therapy or even anti-sera?
So let me tell you the good points and the bad points. The good news is that in the preclinical studies, and that's been done, I believe, in nearly 10 such monoclonal antibodies that have been manufactured in the lab, the results have been very good. They've been getting specific antibody which kills the virus or which prevents the attachment. But in the clinic, there aren't that many infectious diseases for which monoclonal antibodies are effective. You've heard both Dr. Vishwakarma and Dr. Pramod tell us that they work for cancer, they work for autoimmune diseases like rheumatoid arthritis, etc. Been tried in Ebola, may work in early cases of Ebola, but there are not that many infective diseases for which this works. So that's not very good news, right? So I think that we really need to wait for the trials to get completed to understand whether this is going to work.
Okay.
But on the positive side, the technology available today for isolating the effective monoclonal antibodies and testing them out in the lab is way more advanced than it was 10 years ago. So we can hope that this is a specific treatment that may work.
Okay.
And I think the time for trying out repurposed drugs is kind of over, in the sense that we have a protocol, as best a protocol as we can have with the repurposed drugs. And now it's time to develop specific antiviral treatment, and this gives us some hope. Okay.
Let me bring in Dr. Ram Vishwakarma here. Dr. Vishwakarma, your views on that comparison, as I was discussing with Dr. Soin there, and the hope that, we might get much more specific results in the weeks or months to come, because now we're looking at the specific aspects of the virus, not the repurposed, options there.
Yeah, so I think all three advanced monoclonal antibodies, which I described earlier, they are beyond phase 2, and 2 of them are actually phase 2, phase 3 stage. That means they have crossed the first barrier of the safety, and they have already crossed the barrier of showing early indication of efficacy. There are 3 major advantages of monoclonal antibodies. These are the best treatment modalities right now in the hand of medicine across the diseases. I believe that. Number 1 advantage is, as far as COVID is concerned, that, like, for example, best of the vaccines will work in, most effective vaccine is considered which works 50%. That's the best that you can in a vaccine. So still you have 50% population which is not amenable to vaccine. Here, if it works in 100% of the patients when they are infected with the virus. And it worked like a magic bullet, like the way a small molecule works. So because it is targeted for a specific event of the biology of the virus, or pathology of the virus. The third major advantage is, like, for example, convalescent plasma or sera, that when you have given plasma, and if you have low titer and patient is not responding, and after say 15 days, if you have to give another dose, you don't have that mostly available to doctors. And here, if you have this, then you can give like a small molecule pill that you take. You can give 2 doses, 3 doses, 4 doses. For example, RA patient keep on receiving lifelong treatment with these monoclonal antibodies. So I think these are the best modalities available to us. And I'm actually most hopeful of coming therapeutic opportunities from monoclonal antibodies because this virus has mutated over its sisters quite a lot. So we need to really look at what are the Achilles heel of this virus. And I think that's where the monoclonal antibodies will work. And the best scenario I foresee is already started happening is the combination of small molecule with monoclonal antibody. That is what is happening. In fact, UK has started a trial this week itself where they are putting two monoclonal antibodies together along with these small molecule combinations. And I think that's a scenario that is available to the clinic, I foresee in a month or 2 months. And like it was said that these trials will come out faster. Production is not a limit, so we are not worried about millions of doses or something. You can produce as much, and luckily, like I said, India is one of the largest producer of monoclonal antibodies. An Indian company produce them and export them in large extent. So I think we are very comfortable situation as far as these regimens are concerned.
Okay, wonderful. Let's take concluding comments from Dr. Pramod here before we wrap this up. Dr. Pramod, your views on the expectations per se, specifically on this line of treatment and the way forward based on what we know right now and what we are working on.
Now, monoclonal antibodies, they have proved very effective in tackling this COVID also in earlier phase trials, as told earlier, in phase 1, phase 2 trials, and now they are in phase 3 trials. And as I told earlier, they attack the virus or they attack the protein or the antigen against a particular antigen, and we can combine the monoclonal antibodies also as we do it in cancer. We can use two monoclonal antibodies together. That is like immuno-oncology. In immuno-oncology means where we give immunotherapy, we sometimes use two monoclonal antibodies together. So we may use two monoclonal antibodies or one monoclonal antibody and some IL-6 inhibitor or maybe some other substance which attacks the virus load, viral load. And how viral load? Which attacks the inflammation. So there is inflammation in the lungs which leads to higher mortality and morbidity. So this is what we are going to reduce. And India, being the largest producer of monoclonal antibodies, we hope for a better future in tackling COVID-19.
Okay, there it is. Thank you so much, Dr. Pramod Julka, Dr.Arvinder Soin, as well as Dr. Vishwakarma for sharing your time and views with our viewers out there on this very important line of treatment which is being worked upon, that is monoclonal antibodies. The research is ongoing as well as the clinical trials, and we'll get to know more details in the weeks or months to come, hoping that this will eventually be one very effective form of treatment against COVID-19. We'll come back again tomorrow with a different topic. Till then, you keep watching Rajya Sabha Television, but please do not forget to wear your face mask whenever you step out of your house. Thank you.